Hi, I'm Richard Sever, editor of Cold Spring Harbor Perspectives. With me I have Art Weiss of UCSF and Doreen Cantrell from University of Dundee. Welcome to you both.
Thank you.
Thank you.
Welcome to a sweltering Cold Spring Harbor. I apologise for the weather, but at least it's not raining. Now you'll both be talking later in this meeting about T-cell receptor signaling, and as I understand, Art, you're focusing more on the upstream events, and Doreen further down the pathway. So if we just start with you, Art, can you sort of set the scene for receptor signaling?
Sure.
How does the T-cell receptor differ from a typical growth factor receptor?
Sure. Well, the T-cell receptor, like many receptors in the hematopoietic system that recognize antigen, don't have a catalytic domain, don't have a catalytic function. Instead, all of these receptors generally separate their ligand binding subunits from invariant subunits that couple to intracellular signaling pathways. And in particular, antigen receptors use a sequence motif that encompasses about 17 amino acids and has two tyrosines that are spaced by about 12 amino acids from each other. And these two tyrosines become phosphorylated through the action of Src kinases. If these motifs, called ITAMs, are doubly phosphorylated, they recruit a second family of kinases, the SYK family, and in T cells the SYK family member is called ZAP70. Both of these kinases have tandem SH2 domains, and the two SH2 domains bind to the doubly phosphorylated ITAM with a fairly high affinity, measured in the nanomolar range, which is quite unusual, but by incorporating two SH2 domains and two tyrosines, one gains a lot of specificity as well as affinity. But the initiating kinase is the Src family kinase.
So the downstream signals are funneled through ZAP70 and T cells, but what's controlling the activity of the Src kinases that you refer to, and how many of them are there?
So there are 8 Src family members, and they have 2 sites of tyrosine phosphorylation. There's a tyrosine in the activation loop of all Src kinases, and like other kinases, when this tyrosine is phosphorylated, it stabilizes the active conformation of the kinase. But the other tyrosine is really the focus of what I'll be talking about. It's a tyrosine at the C-terminus. It's the tyrosine that was deleted in V-Src, making V-Src an oncogene. So it's a negative regulatory site of tyrosine phosphorylation. It's present in all the Src kinases, and that site is phosphorylated by another kinase that looks like a Src kinase but not anchored to the membrane, and that kinase is called CSK.
Okay, and what's controlling its activity?
So CSK gets recruited to the membrane via its SH2 domain. And there are a variety of adapters that are tyrosine phosphorylated that recruit CSK, but in truth, we don't have a really good understanding of how one regulates the dynamic function of CSK. How much is at the membrane? Is it kicked off during receptor stimulation? So that's a little bit of the direction of what the work we're doing is all about.
And I understand, unlike a growth factor receptor, that you have co-stimulatory molecules that feed in to ensure you get the correct level of signal. Are they working through these Src family kinases as well?
Well, so in T cells, you have CD4 and CD8. So CD4 and CD8 are co-receptors that recognize class 2 MHC molecules or class 1 MHC molecules.
So these are the molecules that are binding the antigen on the
That's right.
surface.
They're the peptide-presenting molecules to the T-cell receptor. And by incorporating these co-receptors into recognition, they bring LCK into close proximity of the T-cell receptor. LCK likes to phosphorylate the ITAM motifs within the invariant chains of the receptor, and that triggers activation.
Right, and as I understand it, it's not an all or nothing event. There's different levels of activation that affect the signals downstream that we'll talk about in a minute with Doreen. Is that correct?
Well, so you're really asking whether signaling can be analog or digital.
Exactly.
Clearly there has to be some threshold that you have to cross in order to generate any signal at all to influence downstream events. But there's probably dynamic signaling occurring all the time, even without a ligand present.
So, and that threshold, is that something that would be set at different levels and in different types of T cell?
Well, that's something we'd like to understand. We don't really understand it well. We do know that in some circumstances of anergy, say, or in T regulatory cells, they're relatively resistant to antigen receptor signaling, so that they don't mediate the typical downstream signaling events that you see in a naive T cell.
And in your work on the kinases that you'll be talking about later, what's the experimental approach that you're taking?
So what we were interested in is how do you set the balance of phosphorylation of that negative regulatory site? That is, what dictates its basal level of phosphorylation, and does it get modified during activation? So opposing the action of CSK is a very abundant transmembrane tyrosine phosphatase called CD45. There are about 100,000 to 500,000 molecules per cell. Wow. So it's incredibly abundant. In the absence of CD45, that negative regulatory site is hyperphosphorylated through the unopposed actions of CSK, and LCK is inactive. So CD45 function appears to be constitutive and permissive, constantly dephosphorylating LCK at that site to some level, and CSK phosphorylating that site in turn. And we think that the concerted actions of those two molecules set the threshold for activation. The way we've tested that in the lab most recently is by titrating the level of CD45 using an allelic series of mutants derived from different transgenic T cells, and also by making a genetically controlled inhibitor that's selective for CSK. What we've done is introduced a mutation into CSK in the gatekeeper residue. This is a technology that was pioneered by Kayvon Shokat at UCSF. We then use a bulky inhibitor based on the common inhibitor PP1, to which additional functional groups have been added.
So this is a cellular protein phosphatase.
So this is an inhibitor of CSK, the kinase that phosphorylates.
Right, the PP1.
The PP1 is a general kinase inhibitor.
Okay.
There is a phosphatase called PP1 that's probably confusing you. But so this is just a small molecule inhibitor. It's a competitive ATP antagonist. And because we've created more space in the mutant form of CSK, we can use this inhibitor, a bulkier inhibitor, to inhibit the mutant but not the wild type. In addition, by comparing the actions of the inhibitor in cells expressing the mutant CSK to cells expressing the wild type, we control for off-target effects.
Right.
The bottom line and the surprising result is when we inhibit CSK with this inhibitor, we trigger the TCR signaling pathway all the way down to PLC gamma 1. This is without adding any ligand. So what that really tells us is that CSK is providing the dominant brake or control on the equilibrium state that exists to control the basal state.
Oh, I see.
And so you've got to overcome CSK inhibition in order to trigger the receptor.
Right. So when the brake is lifted, and ultimately this leads to ZAP70 activation, then there's a constellation of adapters and kinases, GTPases involved, and I guess that's where you come in, Doreen. You're going to be talking about one of these kinases further downstream, mTORC1. Is that correct?
That's correct. That's correct.
And one of the things that intrigued me was that you started mentioning control of this kinase by nutrient levels. Could you explain that a little bit?
Yes, mTORC1. It's a horrible name. It stands for mammalian target of rapamycin complex 1 is the name of this kinase, and it's called that because it was identified because it's the target of action of an immunosuppressive drug, rapamycin, which is used a lot in the clinic to sort of help transplant patients not reject their transplants. It's a very evolutionary conserved kinase, and it evolved in the yeast to mediate the ability of yeast to respond to extracellular nutrients, because if you're a yeast, that's what you care about. And so this kinase is activated when cells increase uptake of amino acids, particularly leucine and glutamine, and it's also sensitive to the uptake of glucose as well in cells.
So multicellular organisms have essentially hijacked this whole nutrient
They've basically
pathway.
hijacked this whole nutrient pathway, and what the antigen receptor seems to do, I believe, is control the ability of T cells to take up glucose, glutamine and leucine, and therefore control the activity of this kinase, mTORC1, which then itself goes on to control further aspects of metabolism and control the expression of some key genes that are important for effector T cell function. Genes like interferon gamma, for example, which is a key inflammatory cytokine that you need to fight viral infections.
So multiple aspects of the behavior of the T cell are controlled by this. What sorts of things? I mean, metabolism is one that it seems connected to the whole.
Metabolism is one. It controls the expression of cytokines. As I mentioned, interferon gamma. It controls a key thing that this kinase pathway does in T cells is control what types of chemokine receptors and adhesion receptors that the T cells express, which then determines how these T cells can traffic around the body. So naive T cells have to constantly traffic between the blood and the secondary lymphoid tissue, and then the lymphatic system and back into the blood, and they roughly do, I was taught as an undergraduate that they go around the body 4 times a day. I have no idea if that's true, but I still tell my students that, so they'll spread that to their students. When you become an effector T cell, you have to stop doing that, and you have to move out into the peripheral tissue, because that's where the virally infected cells will be, or the bacterially infected cells. And the ability of T cells to change this migration pattern happens because they change the type of chemokine receptors they express, and they, in simple terms, they lose expression of homing receptors that take them to lymphoid tissue, and they gain expression of receptors that home them to the peripheral tissues. And this kinase, mTORC1, controls the expression of these different chemokine receptors, and it suppresses expression of the naive lymphocyte homing receptors. So as soon as a T cell has activated mTORC, it loses the ability to home back to the lymphoid tissue, and it gains this ability to home out into the peripheral tissues. So that's obviously quite a key switch.
Function. So in this pathway that you're both dissecting, what's downstream of mTORC1? Presumably there's a bunch of transcription factors running gene expression programs. Are these specific to T cells? Are we talking generic transcription factors?
What is immediately downstream of the kinase, the only 2 substrates we know about in T cells actually for this kinase are another kinase called the ribosomal S6 kinase, and also a protein that's involved in control of mRNA translation, which is called 4-ABP1. After that, there's a bit of a black box, and we then hit some transcription factors whose expression is controlled by TOR signaling, and one of them is a transcription factor called hypoxia-inducible factor 1, HIF1 alpha, and also another transcription factor called SHREBBP1 and SHREBBP2. And the expression of these transcription factors is controlled by TOR signaling, and they then go on to control expression of lots of different genes. We don't fully know all their direct targets or their indirect targets, but we know roughly. One example of a gene whose expression is controlled by HIF1 alpha in cytotoxic T cells is perforin, which is the pore-forming molecule that cytotoxic T cells secrete to lyse virally infected cells. That the expression of that seems to be a direct response of the level of HIF1 alpha that a T cell will actually express.
And transcription factors that control cell division as well?
No, not particularly. No, not particularly. Actually, if you inhibit mTORC1, you don't really impinge on the ability of T cells to proliferate. You slightly slow them down. They may do 3.5 divisions a day as opposed to 4, but it's not much of a slowdown. You affect their ability to differentiate and traffic.
Right.
So they still proliferate reasonably well without TOR or without the HIF signaling pathway.
And when Art was talking earlier on about the notion that these signals are continuous and there's something like CSK setting the levels, do you see this at your level in the pathway, so to speak? Is there a binary effect are we just activating mTORC1, or are there different settings?
Actually has been pretty hard to look at. I believe the effect is quite binary, actually. You certainly see in the first time points of T cell activation that a cell will either choose or choose not to activate the mTORC1 pathway, and when it activates it, it fully activates it. And then it doesn't. So I think we've reached below, we reached the threshold a bit, so it's once the T cell receptor switch has decided to go in that cell, then it will activate the pathway. And there doesn't seem, in terms of T cell receptor signaling, any analog nature to that switch. It seems quite digital. Quite
binary, right. I mean, one of the things that we always talk about in signaling is feedback mechanisms, and is there a level at which the kinds of downstream signals that you've been looking at feed back into the kinases around the T cell receptor?
Well, there's some evidence for CSK that there's some feedback. One of the adapter molecules it binds to has a pH domain, in particular DOC1 and DOC2, and that gets recruited to the membrane with higher levels of some of the phosphatidylinositols. So if you generate PIP3, for instance, you'll recruit more DOC1 or DOC2 to the membrane, and that may then allow more CSK to be localized to the membrane as a means of turning off signaling.
That's interesting. And in that sense, where do you see the next stage for looking at the connections within the T cell receptor complex? Is it? Do you feel that it's nearly all worked out? Or?
Not at all. We still don't understand how the initial ligand overcomes the threshold. What is the nature of that threshold? I think we can now manipulate the threshold, but what is it? What's the nature of the signal that's necessary to overcome it? And some of that is going to be approachable through genetics and inhibitors, but I think we're also going to have to use more sophisticated imaging techniques.
I mean, is that in any way affected by the nature of the peptide that's being presented?
Sure, absolutely. And I think in the signaling field in general, we've been using sledgehammers. In the lipid bilayer system, we use concentrations of agonists that are never achievable in cells. Typically in my lab, to do some of these studies, we use antibodies, high affinity antibodies, to stimulate the receptor. So we're really looking at the extreme of signaling to get a sense of what's going on, and I think we need more subtle probes to look at more physiologic responses.
And is that also an aspiration for you in events further downstream?
Yeah, absolutely, because although we know a lot about what mTORC does, we still don't actually know the full range of substrates for this kinase. We know of 2, but we're sure there's going to be a lot more. What I do know is there's been a very beautiful paper from David Sabatini showing the substrates for this kinase in HeLa cells. Almost none of those proteins are actually expressed in T cells. So I know that there's a lot to find out, and if there's a lot of other substrates in that cell type, then we're likely to have some possibly lymphocyte-specific substrates for this kinase. I think that's one of the messages we'd have, that you can have an evolutionary conserved kinase, but the substrates may be cell type specific. And certainly sometimes I sense disbelief when I tell people who work in fibroblasts what I think mTORC1 does, because they know it doesn't work like that in their cell system. So they think we're wrong. Of course, I think they're wrong. And we end up arguing. But I think the answer is we're both right, and just the kinases are going to have different substrates. And I think that's a general message, actually, of a lot of the kinases that act downstream of the T cell receptor, the serine threonine kinases, we've all been measuring the activity of the MAP kinases for years, but I don't think any of us could put our hand on our heart and say what a substrate is for the MAP kinases, yet it's almost the only signal that most immunologists measure in terms of a kinase. We still don't really understand all the targets for calcium signaling in T cells. We talk about calcineurin, phosphatase is a main target, but that's it, and that cannot be the whole answer. So there's still huge gaps
A lot of work
to fill in, and interesting gaps that when we fill them, we might have some interesting new drug targets to help us.
Well, it sounds like there's a lot of work to be done. Good luck in your search for something lighter than a sledgehammer, and good luck in the search for new substrates. It's been very nice talking to you both.
Thank you.
Okay, thank you.
Thanks.
Thank you.